Okay, so last time we were discussing about Human Genome Project. So first we discussed about the definition of Human Genome Project. Now you know Human Genome Project, it is an international scientific research project, and it had a primary goal of determining the sequence of chemical base pairs that make up the DNA, and it also had the primary goal of identifying and mapping the genes which are present in this DNA, and they had to study the structure and the function of the gene. So we have written that definition. So today we will discuss where this Human Genome Project was first started. This Human Genome Project, it was officially launched in USA in 1990. Who founded this project in USA? It was founded by the US, United States National Institute of Health, and US Department of Energy. Department of Energy. So Human Genome Project, it was formally founded by US National Institute of Health and US Department of Energy in 1990. They estimated this project will cost $3 billion. The estimated cost of this project was $3 billion. And they estimated that the project may be completed it may take 15 years. So the project may be completed within 15 years. So the project was formally founded in USA in 1990 by US National Institute of Health and Department of Energy. And the estimated cost for Human Genome Project at that time was $3 billion, and the estimated time was 15 years. At that time, US sought international cooperation. US sought international cooperation. So other international communities like UK, Japan, France, Germany, China, India, Pakistan, etc. They also joined in this Human Genome Project. So because of participation of many other countries, this project was given the status of International Scientific Research Project. This project was designated as International Scientific Research Project because in addition to USA, other countries also participated in this project. Countries like United Kingdom, Japan, France, Germany, China, India, Pakistan, they also participated in this Human Genome Project. This Human Genome Project, it is known as a mega project. Why it is known as a mega project? Because they had to sequence the entire human genome. They had to sequence entire human genome. And the cost was $3 billion, $3 billion US dollars. $3 billion US dollars. So it was a huge project. It was a very large project which had a very huge cost, $3 billion US dollars. And they had to sequence approximately 3 billion base pairs. 3 billion base pairs of a haploid genome. In diploid genome, there will be 6 billion base pairs. So they had to sequence these 6 billion base pairs of diploid genome. And it would have cost them $6 billion for sequencing diploid genome. So they first sequenced the haploid genome, which involved the cost of $3 billion. So that means they had to spend $1 for sequencing one base pair. There are 3 billion base pairs in haploid genome, and the cost was $3 billion US dollars. So that means one base pair cost them $1, right? So for this reason, it was known as a mega project. It was known as a mega project. Sometimes I have seen a question paper where it was asked to explain why Human Genome Project is known as a mega project. A question was asked in a question paper, why Human Genome Project is known as a mega project. So what is the answer? The answer is that this Human Genome Project, it had a huge cost, 3 billion US dollars, and they had to sequence 3 billion base pairs of a haploid genome. If we talk about diploid genome, then they had to sequence 6 billion base pairs. In diploid genome, there will be 6 billion base pairs, and the cost will be $6 billion, right? Because one base pair costs them $1. So for that reason, it is known as a mega project. Why it is known as International Scientific Research Project? Because in addition to USA, other countries like UK, Japan, France, Germany, China, India, Pakistan, Australia, they also participated in the project. So that made it an international scientific research project, right? So this is how the Human Genome Project was started. As the Genome Project was launched, scientists in different countries, they started working out the chemical sequence of chemical base pairs of this human genome. They started to analyze the sequence of chemical base pairs. And the first rough draft of Human Genome Project, first rough draft of Human Genome Project was prepared by Genome Bioinformatics Group of University of California, Santa Cruz. So when the sequencing started, and all the countries they made their contribution, then all the data was compiled together by Genome Bioinformatics Group. All the data was compiled together by Genome Bioinformatics Group of University of California, Santa Cruz. And this group was led by Jim Kent. So under the leadership of Jim Kent was the head of this group. Under the leadership of Jim Kent, Genome Bioinformatics Group of University of California, Santa Cruz, they compiled the first rough draft of Human Genome Project in 2000. When the first rough draft of Human Genome Project was compiled in 2000 by this Genome Bioinformatics Group of University of California, Santa Cruz, then it was announced in the media by then US President Bill Clinton and then British Chief Minister, British Prime Minister. I think it was Tony Blair. So Bill Clinton and Tony Blair, they jointly made the announcement that the first rough draft of the Human Genome Project has been completed, right? Then during this time, there was a private company. In this time, there was a private company which was led by a scientist, Craig Venter. They also launched a Human Genome Project. Craig Venter and his firm, he had a firm, and his firm Celera Genomics also launched a Human Genome. They also launched a Human Genome Project privately. The project which we were discussing so far was launched by US government. But this project, which was launched by Craig Venter and his firm Celera Genomics, it was a private company, and they also launched the Human Genome Project. They also made the estimated cost $3 billion. They also made the estimated cost of $3 billion. And surprisingly, in 2001, they also published. They also published the first rough draft. They also prepared. They also prepared the first rough draft. Prepared first rough draft. Then, in the same year, 2001, both the drafts of both the drafts they were published together as a joint publication. Joint publication of the Human Genome Project was made in 2001. So one publication was contributed by the official US Human Genome Project. Another publication was prepared by Craig Venter and his firm Celera Genomics. But both the publications were jointly published in 2001, right? Then in 2003, 2005, improved drafts were published. Drafts were published by both the groups. In 2003, 2005, improved drafts were published by both the groups. Then in 2006, last sequence of the last chromosome was published. In 2006, the sequence of last chromosome was published. So in 2006, it was declared that the human genome is now formally complete, right? So when the first rough draft was made, first rough draft was prepared in 2000 by Genome Bioinformatics Group of University of California, Santa Cruz. Then Craig Venter and his firm Celera Genomics, they also launched the Human Genome Project with an estimated cost of $3 billion. They also prepared their first rough draft in 2001. Then in 2001, both the drafts were published together in the form of a joint publication. Then both the groups made improved drafts in 2003, 2005, 2000. In 2003 and 2005, improved drafts, any area which was not sequenced so far. Those areas were sequenced and improvised drafts were prepared in 2003, 2005. In 2006, the sequence of last chromosome was also published. And in 2006, it was announced that Human Genome Project is complete, right? It was announced that Human Genome is complete. But still, Human Genome Project was not complete, 100% complete. Only 92.3% of the Human Genome Project, even after the last draft, even after the sequencing of the last chromosome, is considered to be complete. Only 92.3% of the human genome has been sequenced, even after the publication of the last chromosome. 92.3% human genome has been sequenced. So even after the publication of the sequence of the last chromosome, only 92.3% of the human genome has been sequenced. So that means there are certain areas which had remained unsequenced. What were those areas which could not be sequenced at that time? So first, there are centromeres. Centromeres, they have highly repetitive sequences, repetitive base sequences, base pairs. So these centromeres, they have not been fully sequenced. They have not been fully sequenced. Similarly, another area is telomeres. Telomeres, they are also unsequenced. They could not sequence them by the available sequencing techniques, right? The available sequencing techniques are not sufficient enough to completely sequence centromeres and telomeres. So both the groups said that we have to wait for improvement in sequencing analysis, improvement in sequencing technologies, right? Our current technology of sequencing is not sufficient enough to sequence centromeres and telomeres. Similarly, there are certain loci having members of multi-gene family are yet unsequenced. There are certain locations in the DNA, some genes work together in the form of a gene family. So those regions where some genes are clustered together and work together in the form of a gene family, those regions, those loci, they are yet unsequenced. So only 92.3% of the human genome has been sequenced so far. These regions of the human genome, centromeres, telomeres, and loci having members of multi-gene families, they are yet unsequenced. So we cannot say human genome project is 100% complete. Human genome project has been completed, but only up to 92.3%. And these areas are yet to be sequenced, right? Now, what were the key findings of this human genome project? They have officially now closed this human genome project. It has been declared officially complete, but only up to 92.3%. What are the findings of this human genome project? What were their key findings? We have to discuss what were the key findings of human genome project. Key findings of human genome project. So the first important finding was that human genome consists of 20,500 genes. It was a shocking news at that time. There were estimates that human genome may contain this 1 million genes. Most of the scientists believed that human genome is formed of a million genes. Some were saying no, there may be at least 50,000 genes. But finally, it was disclosed that human genome is only formed of 20,500 genes. The same range as in mice. In mice, there are also these 20,500 genes, and twice that of roundworms. So it was a shocking news at that time. Most of the scientists believed that there may be approximately 1 lakh genes present in human genome. But when the human genome project was completed, it was finally declared that there are only just 20,500 genes. The same range as in mice. जितने हमारे genes हैं उतने ही mice में भी होते हैं. The same range as in mice. And twice that of roundworms. So roundworms, they have got some 10,000 genes, and we have double the number of genes than roundworms. This was the first important key finding. And what was the second important key finding? It was that 1.1 to 1.4% of genomic sequences only coded for proteins. So we have 3 million base pairs in our haploid genome. We have 3 billion base pairs in our haploid genome. Out of those 3 billion base pairs, only 1.1 to 1.4% of genomic sequences they code for protein. Or we can say genes make only 5% of entire genome of entire genome of entire genomic sequences. Genomic sequence. So genes make only 5% of the entire genomic sequences. Then what is the rest remaining DNA? What are the remaining base sequences? At first it was said that the remaining DNA is junk DNA. When they came to know that genes they make only 5% of the entire human genome, right? Then they were asked what about the remaining part of the genome? So first it was said that the remaining DNA, that is junk DNA. But later on it was found that this remaining DNA is not junk DNA. It has got some other regulatory functions. It has got some other functions. But only 5% of the entire genome is made of genes, right? So we say 1.1 to 1.4% of the genome sequences only code for proteins. So we can say genes make only 5% of the entire human genome. Only 7% protein families appear to be vertebrate specific. Vertebrate specific. The proteins which were coded by the genes, only 7% of those proteins. That means the proteins which our body makes, only 7% of those proteins are only found in vertebrates. The remaining proteins we share with non-vertebrates as well. Do you understand what I'm saying? Only 7% protein families appear to be vertebrate specific. That means all the proteins which were formed in our body by our genes, only 7% of them are only contained in shared among the vertebrates. The remaining proteins, 93% of our proteins, we share them with invertebrates as well, right? So 7% protein families appear to be vertebrate specific. Only 7% of the proteins are shared only among vertebrates. And the remaining proteins, 93% remaining proteins, are shared with invertebrates or non-vertebrates as well, right? Then another key finding of the Human Genome Project was that human genome has more segmental duplications. Human genome has more segmental duplication. Segmental duplication as compared to other mammals. So that means our DNA is highly repetitive. Segmental duplication means this is the segment of DNA. In this DNA, there is a sequence of base pairs. Particular sequence of base pairs. This sequence of base pairs is present here. Then it is repeated again here. The same sequence of base pairs is then repeated again here. So a particular sequence of the base pairs is repeated again and again. So these repeats of a particular sequence of base pairs again and again in DNA, that is known as segmental duplication. So they found that human genome has more segmental duplication as compared to other mammals. So these were some important findings of the Human Genome Project. So what was the first important finding of the Human Genome Project? That human genome is only formed of 20,500 genes. The same range as in mice. We are much larger than mice. Mice show up mice Mice and human beings, they have got the same number of genes, 20,500. And twice that of roundworms. Roundworms go ascaris. Ascaris is 10,000 gene. We have got some 20,500, right? So this was the first important. This was the shocking discovery. Then what was the second finding? 1.1 to 1.4% of genome sequences only. 1.1 to 1.4% of genomic sequences only coded for protein. So the remaining sequences of the genes, they did not code for protein. So in other words, we say genes make only 5% of the entire human genome. The remaining part of the human genome is not having genes in it. Genes make only 5% of the entire human genome, right? Then another important discovery was that only 7% protein families, are vertebrate specific. The remaining only 7% protein families appear to be vertebrate specific. So only 7% of the protein families, 7% of the protein families, they are shared only among vertebrates. The remaining proteins are also shared with non-vertebrates. And the last finding which we are discussing here is human genome has more segmental duplications as compared to other mammals. So in our DNA, there are certain regions where there are certain base pairs repeated again and again. So these regions of DNA where a specific sequence of base pairs is repeated again and again, that is known as a segmental duplication. And these segmental duplications, they are very common in, they are very, they are very more, they are repeated again and again. They are very common in human genome as compared to the genome of other mammals, right? So segmental duplications are more in human DNA as compared to other mammalian DNA. So these were the key findings of Human Genome Project. So that is all about Human Genome Project. So what you have to remember, this question is mostly asked in the examinations. So what you have to remember, how you have to remember it. First of all, you have to remember its definition, right? Then you have to remember its history, when it was started, where it was started, what was its estimated cost, what was the estimated time, right? You have to remember that. After that, you have to remember when the first rough draft was compiled, right? You also have to remember why it is known as International Scientific Research Project or why it is known as Mega Project. I have told you that. Then when the first rough draft was compiled, who compiled the first rough draft, right? And when? Then this project was started, launched privately by Craig Venter and his firm Celera Genomics in 1998. They also made their first rough draft in 2001. Then in the 2001, both the drafts were published together as a joint publication. Then in 2003, 2005, improved drafts were published, right? Then in 2006, the sequence of last chromosome was published in journal Nature, right? So that was the milestone. And at that time, Human Genome Project was declared as complete. Even after they declared Human Genome Project as complete in 2006, but there were certain regions in the human genome whose had remained incomplete. What were those regions where the sequencing had remained incomplete? Those were centromeres, telomeres, and certain loci having members of the multigene family. So in aggregate, 92.3% of the human genome has been sequenced, and the remaining part still remains incomplete, unsequenced. Why it still remains unsequenced? Because our current sequencing technology is not sufficient enough for sequencing centromeres, telomeres, and members of multigene families, right? So we have to wait for the improvement in the sequencing technique, sequencing analysis, right? So that after that, you have to remember what are the key findings of the Human Genome Project. So as I mentioned just now, first of all, you have to remember that it was found that human genome is only formed of 20,500 genes, the same range as in mice, and twice that of roundworms. Twice that of roundworms. Then you have to remember only one point. only 1.1 to 1.4% of genomic sequences coded for proteins. Or you can say genes make only 5% of the entire human genome, right? Then only 7% protein families appear to be vertebrate specific. Only 7% protein families are shared only among vertebrates. The remaining protein families, they are also shared among invertebrates, right? Then you have to remember that this human genome has more segmental duplication as compared to other mammals. So in our DNA, there are certain regions where a specific base sequence is repeated again and again. These regions of the DNA where a base sequence is repeated again and again, we call them as segmental duplications. They are much more common in human DNA as compared to other mammalian DNA. So that is all about the Human Genome Project. So that is all for today, right? Inshallah, next lecture we will discuss DNA fingerprinting. In next lecture we are going to discuss DNA fingerprinting. Okay, thank you. Thank you.